Alright, today our discussion will be starting from chapter 2, cells. Yesterday we finished off chapter 1, so we are starting off with chapter 2 today. Alright, let's start. Chapter 2, cells. Alright, great. So, cell. If I talk about cell, can I say cell is the basic structural and functional unit of life, right? So, it is the basic, it is, one second. Yeah, it is the basic structural and functional unit of life. All organisms are made up of cells, and obviously, we cannot see cells with our bare eyes because they're very small. So, we use a microscope for that, and there are two types of microscope. We have light microscope and electron microscope. Light microscope and electron microscope. Light microscope gives us an image up to 1500 times magnified. Electron microscope gives it to approximately 500,000 magnified. So, electron microscope is obviously better with magnification and all, but resolution and all, there are perks for both light and electron. All right, moving on to your organelles in your animal and plant cell. So let's talk about animal and plant cell. First, we'll talk about all the things that are common in plant and animal. First, let's talk about cell membrane. It's a very thin layer of protein and fat, and it is important because it controls what enters or leaves the cell. So I can say it is a partially permeable membrane that controls what enters or leaves the cell. Partially permeable, selectively permeable, semi-permeable, you can call it anything. All three names. Partially permeable membrane that controls what enters or leaves the cell. Right. It also basically separates the contents of the cell from its surroundings. It separates the contents of the cell from its surroundings. All right. Secondly, let's talk about cytoplasm. Cytoplasm is basically a clear jelly and it's nearly all water. Okay, 70% of the cells are made up of water. Okay, it's nearly all water. Alright, so as you can see, jelly-like substance. And is nearly all water. All water. All right. Oh, then it contains many substances dissolved in it. It contains many dissolved substances. And different, metabolic reactions take place in the cytoplasm. The site of metabolic reactions. We also call them cytosolic reactions because cytoplasm is also called a cytosol. It's called the site of metabolic reactions. Alright. Then let's talk about nucleus. So this is where your genetic information is stored. Alright, information is kept on the chromosomes, and chromosomes are made up of DNA. Alright, so chromosomes are very long and so thin that you cannot basically see them even by using your electron microscope. Okay, and they can be only seen at the time of cell division because they come short and thick. So at this, at that time they can be seen using your even your light microscope. Okay, so you can say place where genetic information is stored. Okay, information is kept on chromosome, which are made up of DNA. Okay, chromosomes are very long and thin. That they're not easily visible even by using electron microscope. Not visible. They can be seen at a time of cell division since at that time they are short and thick. They can be easily seen using a light microscope at the time of cell division since chromosomes are short and thick. Alright. Ribosomes. Ribosomes are basically tiny structures that are found in all plants and animal cells, and they can be only seen using an electron microscope. They're very small. Okay. Only seen using an electron microscope. Site of protein synthesis. Okay. Now, so the instructions on the DNA molecules are used to link together long chains of amino acid into a particular sequence. Okay, and that makes protein. So this is why protein synthesis takes place here. DNA sequence comes from the the instructions come from the nucleus to the ribosomes. We're going to talk about this in detail, inshallah, later on the chapter. Okay, now mitochondria. So mitochondria is the site of aerobic respiration, and this is how energy is released from glucose. Okay, and number of mitochondria basically depend on the activity of the cell, right? If the cell is very active, there are a number of mitochondria. Site of aerobic respiration. Where glucose is broken down to release energy. And number of mitochondria, like I said, depends on the activity of the cell. Okay, energy requirement of the cell. And muscle cells have a lot of mitochondria. All right, liver cells. All right. Now let's talk about the other organelles. Firstly, let's talk about vacuole. Now, vacuole is present in both, but I took it at the last because there are size differences. Okay. So, animal cells have a small and temporary vacuole. Have small and temporary vacuole. Plant cells have large and permanent vacuole. Okay, so what is a vacuole exactly? It's a fluid-filled space inside a cell that is surrounded by its own membrane. So it is a fluid-filled space. And usually in plants, it contains a solution of sugars and other substances that is called your cell sap. Okay, it contains cell sap.
And what is cell sap?
Cell sap is basically a solution of sugars and other substances. All right.
Okay, ma'am.
So solution of sugars and other substances. Is everyone clear up till here on all the organelles we have discussed till now?
Yes.
Yes, ma'am.
Yes,
Ryan?
miss.
Oh, Ibrahim? Zoya?
Yes.
Okay, great. Alright, let's move on. Now let's talk about the two distinctive organelles which animal cells do not have, but plant cells have: cell wall and chloroplast. Okay, firstly, cell wall. So all plant cells are surrounded by a cell wall. Okay, so in plant, cell wall is made up of cellulose. Plant cell wall is made up of cellulose. Please remember this. Fungal cell wall is made up of chitin. Plant cell wall is made up of cellulose. Bacterial cell wall is made up of peptidoglycan. Okay, so cellulose basically belongs to a group of polysaccharides. All right, and it basically forms fibers which crisscross on top of one another to form a very strong covering to the cell. Right? Cellulose forms a very strong covering around the cell. very strong covering around the cell. And it basically helps to protect and support the cell. Protect and support the cell. Cell wall absorbs lots of water and swells, which stops the cell from bursting. Okay, so what exactly happens is cell wall is fully permeable. Okay, cell membrane is partially permeable, cell wall is fully permeable, so it allows everything. Now, so when a lot of water enters the cell, what happens? The contents of cell starts pushing against, because cytoplasm is filling up with water. Okay, so they're pushing against on the cell wall, and first they're pushing to the cell membrane, and then cell membrane will push across to the cell wall. Right, so a cell wall with then your cell contents as the swell, the cell wall does not burst. That's the important point. Animal cells can burst because there's no cell wall in them. The cell membrane. Cell membrane can burst. It does not have any cellulose or anything like that. Cell wall cannot burst because it has cellulose. Right? There are spaces, when I said they form crisscross, so there are spaces between fibers. Alright? So it swells and prevents the cell from bursting. Okay, it is completely permeable, or you can say fully permeable. We'll see in the next chapter when we talk about osmosis, and when we'll see how your cell contents push on the cell wall. So okay. Um, what end is the cell? It swells. Alright, which prevents the cell from bursting. We're gonna talk about this in detail in the next chapter. Inshallah. Alright, then second is your chloroplast. So both your cell wall and chloroplast are only found in your plant cell. Okay. So chloroplast contains a green color pigment that is called your chlorophyll. And chlorophyll is the pigment that absorbs light. Okay, it's not chloroplast absorbing light, it's chlorophyll that absorbs light. So I will say contains green color pigment called chlorophyll that absorbs sunlight. Green colored pigment. Which absorbs sunlight. Okay. And then this energy is then used to make food by the process of photosynthesis. Okay. They also contain, starch grains which have been, made by photosynthesis. Whatever is stored. All right. Are all the organelles clear?
Yes, ma'am.
Yes, miss.
Alright, great. Please quickly note them down, and then we're gonna talk about your bacterial cell. We have discussed it in the previous chapter as well, so we're gonna just quickly go over it. Quickly note this down. Tell me
Miss,
when to scroll. Yes.
the chloroplast contains chlorophyll as well as the starch grains.
Yes. See, starch grains have nothing to do with chlorophyll. See, starch grains are basically the stored form. Whatever glucose is left over, like it's not used, chloroplast stores it. Okay, in the form of starch. There's some starch grains as well. All right.
Okay.
So quickly note this down, and I'm gonna move on.
I'm done, ma'am.
All right, everyone's done. Should I scroll down?
Please show vacuum.
Vacuum. Okay. Please note this down quickly. Alright, done.
Yes, ma'am.
I'm done.
Alright, then let's move on. Zoya, Ahmed, Ibrahim. Alright, let's move on. Okay, then let's move on. 1 second. Alright, bacterial cell. Yesterday we saw the structure of bacterial cell. Let me paste a diagram again so that it's easier for you guys. So, a bacterial cell is a unicellular organism. Bacteria is a unicellular organism. You guys know that. Okay. Bacterial cell wall is made up of peptidoglycan, right? Unlike your plant cell wall, which is made up of cellulose, fungal cell wall is made up of chitin. Okay. Alright. So, again, same function of your cell wall to protect it, to protect the cell, okay, and provide, structural support. Okay. Again, it stops it from bursting and all. Partially permeable cell membrane, same. Alright. Only thing different they have is plasmids, okay, that contain the bacterium genes. Okay, cytoplasm ribosome, same function as of your animal plant cell. Alright, but instead of chromosome, you basically have DNA. Alright, and you can say let's write regular DNA. Okay, fine, it's okay. It's a loop of DNA basically. Okay, it's called the bacterial chromosome. They have no nucleus, so this is what sends instructions to the ribosome, and they have a flagellum, a slime capsule. Again, these two features are not important for all bacteria to have, but a typical bacteria contains these. Alright, moving on. Now let's talk about the specialized cells. Typical bacteria is not important that they have this flagella and what else? Capsule. Okay, it's not important. Everyone, all bacteria have it. There are other features, pili as well. Okay, and some things as well that some bacteria have, some do not have. Pili is basically used for conjugation. It's not important as of now. Okay, let's talk about specialized cells. Alright. So, multicellular organisms, you know that they contain millions of cells, and almost all of the cells have the same features, but most of the cells have a particular function to perform, and the structure is modified to help them carry out the function efficiently, right? I mean, all cells are cells. All cells have the same features, but again, their functions are different, and the structures have been modified to perform those functions efficiently. Okay, so these type of cells are called specialized cells. All right, so also all cells have same features, but most cells have different functions. And their structures are modified to perform those functions efficiently. So these type of cells are called your specialized cells. So these cells are specialized cells. Now cells make up tissues. Let's talk about tissues. Tissues are what? A group of cells working together to perform a specific function. Working together to perform a specific function. Then group of tissues make up organs. A group of tissues that works together to perform a function together. That works together to perform a function. To perform a function together. Next, when you know a group of organs is working together, it will be called as your organ system. So you will say different organs working together to perform function and run a body. Working together. And run the body. Alright. Well, let's talk about the size of specimen. Right? Sometimes the specimens are so small that you obviously need microscopes to look at them. You cannot use your bare eyes. And whenever you are looking at the specimen in the microscope, you need to be able to tell how much are you magnifying that specific specimen. The formula for magnification is image size upon actual size. Just remember this triangle. Let me just make this triangle. Right, I on the top, image size, and I am. Okay, this is multiplication, and this line shows division. Okay. The center line is your multiplication, the top line is your division. So I is image size, A is actual size, M is magnification. Whenever you're writing the magnification, always remember to write the value with a times sign, like this many times the magnification, the image is magnified, and always give your answer in whole number. Okay, for example, if your magnification comes out to be 1333.3, this will be incorrect. You will write that it is magnified up to 1300 times. This is right. Always make sure to keep the unit same. These two conversions are very important. Please learn these. 1 micrometers is equal to 1 exponent negative 6 meters, and 1 meter is 10 to power 6 micrometer. Okay, you can convert it to centimeter as well. Just remember any one, and rest of it you can do it on your own. Is this clear? Any questions?
No questions.
Centimeter to micrometer is times 1000. Write this as well. Sorry, 10,000. Astaghfirullah, not 1000. So times 10,000 centimeter. And vice versa will be divided. Yes. Alright. That's all for this chapter. Note this down. Then we are gonna move on to chapter 3. Different types of specialized cells. These specialized cells are very simple. You don't really learn all of them. Like there's so many. Like what are the types of specialized cells? Red blood cells are specialized cells. Nerve cells are specialized cells. Ciliated cells are specialized cells. I mean, how will you learn? There are so many. It's pretty simple. Okay. Muscle cells are specialized cells. Platelets. Alright, so is this clear? You don't need to like actually learn all of them. Yeah, so you can, just for example, they will not write all of them. So I gave you 5 examples right now. Okay, but you cannot learn all of them, okay? Because there's no possibility of, of specialized cells. Because so many, like you cannot, learn all of them. Just remember, all types of cells are specialized, like blood cells, muscle cells, villi, your epithelial cells, and what else? White blood cells, platelets, neurons. Okay, Ryan, is this clear? Did anyone else any question? See, then learn those. Then okay, fine. See, I don't think they will ask you to like write all the specialized cells names. Right, and I think this is something that is understood and you don't actually need to learn it. See, we cannot learn. There are so many things to learn that you are gonna see, inshallah, later on in the syllabus. That this is very, it will be very hectic. Okay, so I think it's understood. The concept is understood. Anything that has its own specific function is a specialized cell. Now, I don't think you will have to make a list of it and learn it separate. It's not something considered separate that I will sit and don't learn this. All right. So I hope this is clear. Anyone else? Any questions?
Everything clear, miss.
Okay, quickly write this down. Then we're gonna move on to the next chapter.
Miss, in the conversions that you have written, this is a micrometer, right?
Yes, micrometer.
Oh, thanks.
No problem. All right, done.
Yes.
Yes, ma'am.
I'm done.
I'm also
All right. All right, that's great then. So let's start off with the new chapter. Chapter 3, that is movement into and out of cells. movement into and out of cells. Alright, so we need substances all around our body, and substances need to move into and out of cells. Okay, for example, glucose needs to enter the cell, oxygen needs to enter the cell for breaking down of glucose. Okay, and then carbon dioxide and water needs to leave the cell. Okay, so particles are always in motion, and as the temperature increases, your particles motion also increases. Okay, and they move faster, right, because they gain kinetic energy. All right, so movement of substances basically has 2-3 ways in which your particles can move. Okay, substances can move. You have diffusion, osmosis, and active transport. Firstly, we'll be talking about diffusion. So I will say diffusion is the movement of particles from an area of higher concentration to an area of lower concentration. Okay, down the concentration gradient. Concentration gradient. Okay, so if I draw a graph, they're gonna be moving down the concentration gradient. Over here, you will have low concentration. Over here, there will be high concentration. And this process does not use energy. Alright. So living organisms get many of the substances they need by diffusion, and they also get rid of many substances, many waste products as well, like I just gave the example of respiration. Second we have is osmosis. So I will say movement of particles. Now this is water particles obviously. And in this time, you'll be saying they move from an area of higher water potential to an area of lower water potential. Let's see what exactly water potential is. Let's change the definition. And this time it's water particles from an area of, higher water potential to an area of lower water potential. And down the water potential gradient. Down the water potential gradient. And across the semi-permeable membrane. So, and again, this is also a passive process, does not require energy. Basically, the word water potential means number of free water molecules. So basically, your substance is moving from a dilute solution. Your water particles are moving from dilute to concentrated, because dilute has higher water potential. It will have more free water molecules, and concentrated will have less free water molecules. Number of free water molecules. Is this clear? So your movement of water particles is from dilute to concentrated. Is it clear? Yes or no?
Yes, miss.
Yes.
Good. Dilute is high water potential. Concentrated is low water potential. So you can also say movement from a dilute solution to a concentrated solution down the water potential gradient. But this across a semi-permeable membrane is very important. Don't forget this. Otherwise, there is no difference in diffusion and osmosis. Right? In this, there was concentration gradient. In this, you will have water potential gradient. Just copy this. From high water potential to low water potential. Water potential gradient. High water potential. So basically, just say dilute solution to concentrated. To concentrated. Will they consider rock fish concentration? Not exactly. They in mark schemes they have said that students, except if students use the word concentration. But it's a concentration of water particles, okay? Just use the word water in that, okay? It won't be wrong, but better is to use the word water potential. Alright, okay, great. Now if I talk about osmosis and animal cells. So since animal cells do not have a cell wall, if too much water enters the cell wall, okay, if you place an animal cell into concentrated solution, since there is no cell wall, water will move inside and cell will burst. Firstly, let's place it in pure water, okay? So if animal cell is placed in pure water, water will enter inside the cell. Water will enter inside the cell and cell will burst. Because inside there's more concentration, so water will move from outside to inside. And since there is no cell wall, your cell will burst. If animal cell is placed in concentrated solution, inside is dilute solution, so water will move outside the cell will shrivel. It will basically shrink. In concentrated solution, water will leave the cell and the cell will shrink. Or you can say shrivel as well. Shrivel or use the word shrink. Now, if I talk about plant cell. So plant cell, firstly, it will not burst if you place it in pure water because you have cell wall. We have talked about this in the organelle discussion as well. Okay, it's fully permeable and it will let any molecule pass through it. So plant cell, if you place it in pure water, water will enter inside the cell and it will become turgid. Okay. The plant cell is placed in pure water. Water will enter the cell, but it will not burst because it has a cell wall. If the plant cell and the cell will become turgid. Turgid is normal. Okay, it's not something abnormal. Turgid means it is proper. Okay, turgor pressure is being applied. Turgor pressure is basically the pressure of the contents of the cell that they apply when water enters it. When water enters it. Okay, so turgor pressure helps the plant to stay upright. Okay, it's normal condition to stay upright. On the right, the plant is placed in concentrated solution. Okay. Now your cell will lose water, obviously, because water will move outside the cell because it moves from an area of higher water potential to an area of lower water potential. Let's see then what happens. Then your cell will lose its turgor pressure and it will become flaccid. Concentrated solution. Concentrated solution. And the cell will become flaccid. And if it stays flaccid means it becomes sloppy. Okay, you might have seen leaves, how they become droopy, they wilt. That is flaccid condition. And sometimes it loses so much water that your cell wall, your cell membrane detaches away from the cell wall and the cell becomes plasmolized. If cell loses too much water, cell membrane detaches from cell wall and cell becomes flaccid. Plasmolized, sorry. It detaches away from it and then cell death happens. One second. Last way of moving substances is your active transport. So in that, your particles move against the concentration gradient. Okay, so an active transport, and they require energy because they're moving against the concentration gradient. There is a movement of particles from an area of lower concentration to an area of higher concentration against the concentration gradient. Alright, and using energy. Okay, and at times they also use your carrier protein or protein pumps for transporting large molecules, or protein pump. are also used. You can add using energy in the form of ATP, or you can say using energy from respiration. Just adding the part of energy is very important. So in osmosis, we learned 4 terms. Okay, firstly, it's shriveling or shrinking. Okay, burst is okay, simple, you guys understand. Then we talked about turgid, we talked about flaccid, and then we talked about plasmolized. Is this clear, everyone? Any questions? Why does not a cell
Miss,
burst
what
if
is meant
submerged?
by plasmolized?
Plasmolized means, Zoe, one second, come to your question. Plasmolized means that your cell membrane detaches from the cell wall. Because of lack of water, it starts to detach. Okay.
Yeah.
Zoya, you said, why doesn't a cell burst if you submerge a finger or anything in water? See, you really think your water can enter through your skin? That is just the external part. Water enters when you drink it. Submerging has no thing, no relation with your movement of water in cells. How you think we can transport substances through our skin? Skin is just an external barrier. Alright, is this clear, sir? Any questions? Water enters the cell when you drink it internally. Alright. Okay, great. Please note. Anyone else any question? Let me just make another graph for your active transport as well. Anyone else any questions meanwhile? Yes. No? High to low. This is also dilute to concentrated, and this will be against. Okay, this will be against. This is from low to high. Okay, using energy. Good. Anyone else any questions? Is this clear? All good? All clear?
Yes, miss.
Miss, can you zoom in the active transport?
Yes, sorry, active transport.
Yes, can you zoom in a bit?
Zoom in. Okay, okay. Rest of you, are you like any questions? Rest of you, Umar, Alia, Ryan, all good, all clear, Ahmed, Ibrahim, Saad.
All clear.
Okay. All right, great. Umar, Saad. Okay, great, great. Everyone's clear. All right. You note this down and let me know when you guys are done. So wow, we covered 2 chapters today. Alhamdulillah.
I'm done.
All right, great. So that's all for today. I'm gonna see you guys in the next class. Inshallah, we're gonna start off with chapter 4.
Thank you, miss. Bye.
Allah Hafiz, bye.
Love is.
Allah Hafiz.
Allah Hafiz, ma'am.
Allah Hafiz. Allah Hafiz.